State University. So her lab is working on the characterization of the genes in plant QTL that are involved in broad spectrum disease resistance and biomass accumulations. So today she's talking about Xanthomonas oryzae effector as a tool to identify disease resistance in rice. So please welcome Dr. Jan Leach.
Well, thank you and good morning. I'd like, before I start, to thank the organizers for a very wonderful meeting. Everything has gone so well that it's almost spoiled us, I think, for the future. Today I'd like to talk to you a little bit about TAL effectors and some tools that we're developing that should help in the identification of germplasm that shows resistance to bacterial blight and bacterial leaf streak in the future. And in case I don't get to the last slide, I'd like to point out that my collaborators in this project are Valerie Verder and Lindsay Triplett, who together worked heavily on this project, along with Rene Correll when he was an undergraduate, and then Adam Bogdanov and some folks from his team, now at Cornell University, but used to be at Iowa State. So I'd like to give you a brief history of TAL effectors in rice bacterial blight pathogens. These are effector genes, and I'll give you a little bit more description of them a little later, that are cloned from Xanthomonas oryzae originally. And they were originally shown to be members of a very large gene family with a very unique structure. This central repeat domain dominated our curiosity for years. These genes were responsible for the race-specific recognition by different R genes in rice. So, for example, aviar XA10 conferred resistance on rice with the XA10 gene, and so on. They also were shown to contribute a long time ago to pathogen virulence. That is, if you knock them out, the pathogen loses its ability to colonize the plants. And they're also very important in pathogenic fitness. That is, for the pathogen to survive in the field, these genes, and to infect plants over and over again, these genes are required. And knowing the contribution of these genes to pathogen fitness was several years back showed to enable us to predict the resistance gene durability, and this was work by Nolly Veracruz. So these very interesting genes, the TAL activator-like transcription factors as they're now called, they used to be called AVR genes or AVR BS3 homologs, but these genes encode proteins that are injected into the host plant cell via a type 3 secretion system from the bacterial pathogen into the host plant cell. This system, the type 3 secretion system, is conserved across human pathogens and plant pathogens. They localize to the plant nucleus, and we'll talk a little bit later about how that happens. And once they're in the plant nucleus, they actually control or regulate plant gene functions. So in other words, these bacterial genes have what it takes to control eukaryotic gene function. They can get into a nucleus and control the gene function within the nucleus. These, again, are components of virulence for two important bacterial pathogens of rice: the bacterial blight pathogen, Xanthomonas oryzae, and the bacterial leaf streak pathogen, Xanthomonas oryzicola. The TAL effectors are really interesting because they contain this central repeat domain, but they also contain the nuclear localization signal sequences, which enable them to get into the nucleus. And they have a C-terminal activation domain that's required for plant gene activation. And then this interesting central repeat. We knew this for years, but as one of my friends likes to point out to me in a very kind way, I wasn't bright enough to figure it out. So other people figured this out, and it turns out to be a very exciting and interesting story. And that is that within this central repeat domain, there are two pairs of amino acids at positions 12 and 13, which actually bind to the promoter region on very specific sites in the plant nucleus, and these amino acids and this particular position bind specifically to particular motifs in that region and activate transcription of the genes. So these tau proteins, in other words, activate expression of very specific genes. And today I'm going to talk about a couple of the genes that they target, which are the OS-suite genes. And so for example, AVREX-A7 and PTH-X01 activate the OS11 and 3 genes, and others activate other genes. But they actually bind to these effector binding domains in the nucleus, or the promoter region of the genes, to do that. So a few years back, so that's, for 20 years, that's what we knew. And now, a few years back, finally, Adam Bogdanov's group and Ulabonos's group deciphered the code and figured out exactly how those amino acids in that central repeat domain bound to the effector binding sites. And then, just recently, people from Wolframer's lab have shown that they actually activate these genes called OS-suite genes, some of them, activate in susceptible plants the expression of genes that are involved in susceptibility. They divert nutritional resources away from the plant to the bacteria. So in other words, the bacterial pathogen is telling the plant to feed me. Now, these very same proteins in the evolution of effector-triggered immunity, the host plants have evolved to recognize those proteins, these tau effectors, and respond to them to stop pathogenesis and resist the pathogens. And that's what we had for years measured, was the avirulence properties of these proteins. Well, what you probably have heard about in the last 3 years are how useful these tau effectors are for genome editing or gene editing in plants. And this information, the fact that you can take these effector proteins and use them to modify or change genes in a site-directed way or targeted mutagenesis, has really revolutionized biology. There's a nice summary, a report of how this can be used in plant genomes that's coming out from Adam Bogdanov's lab in plant physiology. It's out now, early ahead of print. But for example, one example that's out there already is from Bing Yang's lab at Iowa State. And he used the TALEN editing properties to modify the promoters of an R gene and actually generate a plant. He makes it so the plant can't recognize the effector, and generated plants that are resistant. And this was published last year. So his talented rice is resistant to disease. So these molecules are very useful, and they've really revolutionized biology. But that's not what I'm going to talk about today. I'm going to talk about other uses for the tau effectors. And I'm going to lead you down the path of how we might be able to use these as tools to identify novel sources of resistance in rice, and also to understand something importantly about the biology of how these proteins affect the host plant. So I'll give you a little more information about the taus. Both Xanthomonas oryzae and oryzae strains harbor large numbers of these taus. So unlike other xanthomonids, which are the pathogens where taus were discovered from, and almost the only pathogens where they're found, the Xanthomonas oryzae's group harbor large numbers. So for example, in the oryzae, the bacterial blight pathogen, in the sequenced genomes we see between 16 and 20 tau effector genes within the genome, so they've amplified. In Oryzicola, Xanthomonas pathovirus Oryzicola, there are more than 26, in fact, way more than even in Xanthomonas oryzae. And Valerie Vigier has worked for a long time with African isolates, and they're quite different. They have fewer tau, but We know the function of only maybe 5 or 6 of these tau in terms of host disease resistance or recognition in the plant or function in disease. So we have many, many tau, but we don't know what they do or if they do anything, if they're just there sitting in the genome. And part of the problem with asking these questions is that because there are so many tau in each of these strains, it complicates the analysis of their function. So if you have 26 tau, you don't know what the interactions between them are. You don't know what the interactions, how they affect the interactions with the plants. So a number of years ago, we had identified in the U.S. a strain that gave symptoms somewhat similar to bacterial blight. And we ignored it for many years. We characterized it, but we ignored it because it was more of a nuisance than a problem. In fact, this strain was so weakly virulent it caused less than 1% yield loss, and it was only found in certain areas of Texas and Louisiana. We called it bacterial blight, and we called it Xanthomonas oryzae at the time, because that was the level of technology that we had. But then, with the advent of genome sequencing and the ability to characterize these things, we knew it was different than the Asian and African strains, but we didn't know exactly what it was. So with the advent of genome sequencing, we were able to sequence a number of strains very quickly, and we found out a lot of very interesting things, but what's most important to my story today is that these U.S. strains, which are weakly virulent, do not contain any tau effectors. They're totally deficient. They don't even have remnants of the genes. There's no footprints, there's no trace of them ever having any tau effectors in their genomes. So we got to thinking that maybe these tau-deficient U.S. strains would be a great platform for investigating the biological roles of tau, for one thing, because you can put a tau in one at a time, and they'd be a great tool for identifying novel sources of resistance. So we first asked the question, do they express and deliver tau into the rice plants? So we expressed in this U.S. strain, which gives a slight water-soaking response on IR24 in the near isogenic line, which has the XA7 gene for resistance. We expressed the AVR XA7, which is a tau gene, a tau effector gene. So on the host with no resistance gene, you get a water-soaking response. And on the host with the XA7 gene, you get a nice hypersensitive response, which suggests that the U.S. strains have a type 3 secretion system which is competent to deliver the effector genes that we can put into it. So it's a great tool for studying. We also then asked, could we monitor the effects? So we knew that certain tau, we already knew that certain tau could increase virulence, or if you knocked them out, they could decrease virulence in other oryzae strains. So we asked if we put a tau effector that was known to increase, and we did this for a number of tau, but I'm giving you one example. It's known to increase virulence to susceptible rice, express that tau in the U.S. strains, and sure enough, we get longer lesions. And Valerie and Renee measured the movement of that effect, the strain expressing the effector through the leaf, and you can see that they detected the transgenic bacteria further down into the leaf. So it increased not only the ability to colonize the plant, but it increased the ability to cause disease symptoms on the plants. And we measured the ability of these bacteria to induce expression of the OS-sweet target, the gene that they're known to target. This is the virulence gene that they're known to target. And sure enough, they do induce, when they harbor that particular effector, they induce the known target, but not when they don't harbor the AVR gene, or the effector. So we stretched this and added a number of other different Xanthomonas oryzae type TAL effectors into this strain and asked if they drove the expression of the targets that they should, and if they also increased virulence. And sure enough, if you put effectors from a number of different strains in, and we show a few here, if they induced an increase in virulence, or if they were important for virulence in their original strain, they also increased the virulence of the U.S. strain. If they did not have an effect on virulence in the original host, they did not increase virulence in the U.S. strain. And in fact, they induced their targets, their off-suite targets, so they induced the susceptibility genes as was expected. So I'm going to give you a little bit of the biology lessons that we learned using this strain before we come around to its use as a tool. So one of the big questions is why does Xanthomonas oryzicola colonize the intercellular spaces, so between the mesophyll cells of a rice plant, and it enters through the stomates? Why doesn't it colonize the vessels like Xanthomonas oryzae? It's a very close relative. Xanthomonas oryzae, the bacterial blight pathogen, it invades through the hydathode water pores, which are on the surface of the leaf, and then it gets into the vessels, and it colonizes the vessels and causes a vascular disease rather than a leaf spot type disease. So we wondered if these TAL effectors were responsible for that biology. And so we inoculated them in different assays, the ones that we use for bacterial leaf streak and the ones that we use for bacterial leaf blight or bacterial blight, to see if we could convert a Xanthomonas oryzicola into a Xanthomonas oryzae, or what could we convert the U.S. strain into. So in this particular experiment, we expressed the TAL targets from Xanthomonas oryzae, the vascular pathogen, in the Xanthomonas oryzicola, the intercellular pathogen. And this is an assay that we use for leaf streak, where we infiltrate the leaves, and then you see a virulent strain will spread out from that site intercellularly, whereas the virulent strain, the avirulent strains stay fairly contained within the spot. And what we saw was that we could increase the virulence of the bacterial leaf streak pathogen, its ability to colonize in this assay, by addition of the effectors from Xanthomonas oryzae. But we could not not engineer with these TAL effectors the bacterial leaf streak pathogen to cause bacterial blight symptoms. So in other words, we couldn't change its pathogenicity profile. We could make it more virulent in its own environment, but we couldn't turn it into a Xanthomonas oryzae. So what about the other direction? So what we did is we took TAL effectors from oryzicola, and these were contributed by our collaborator, Adam Bogdanov, and he had shown that some of these actually do indeed function in virulence for bacterial leaf streak. So we added these TAL effectors from Xanthomonas oryzicola, the leaf streak pathogen, into our U.S. strain and asked, could we convert it into a bacterial leaf streak type pathogen? And you can see here that this TAL2A, which is known to be a contributor to virulence to Xanthomonas oryzicola, did not increase the virulence of our U.S. strain. So we also asked, could we make the Xanthomonas oryzae from the U.S., the weakly virulent strain, have a bacterial leaf streak or bacterial blight phenotype with the Xanthomonas oryzicola TAL? So would they turn it into a bacterial blight type pathogen? And the answer is no. We did not see an increase in virulence in the lesion length assay, which is the bacterial blight assay, by the addition of the Xanthomonas oryzicola TAL. So one of the questions is, well, maybe the U.S. strain cannot deliver the Xanthomonas oryzicola tal effectors. Maybe it's just not competent to deliver them. So because we were collaborating with Adam Bogdanov, he contributed to us a really useful tool. And that is, they had created a transgenic rice where they had put in front of the bacterial blight resistance gene XA27 an engineered promoter that contained 6 different binding sites that were specific to different TAL effectors. So the ones that you'll recognize that I've talked about are PTHX01 and TAL2G. Those are the 2 that I've showed you so far. Well, so we call this the 6-pack, and the 6-pack promoter. So what should happen is, with those 2 effector binding sites in front of the XA27 gene, if we add a TAL to the bacteria and it's delivered into the host plant cell, the TAL should bind to the PTHX01 effector binding site and activate XA27, and we should get a hypersensitive response. So we did that. We put the TAL2G and the PTHX01 in front into individual US strains and asked, do they activate the hypersensitive response? And the answer is yes. When you express the TAL2G, which is the Xanthomonas oryzicola effector, in the US strain, the PTHX01 is a control, it's the Xanthomonas oryzae effector, you actually activate a hypersensitive response. And we actually showed that you also activate the downstream effects. So, our hypothesis is that the U.S. strains are predisposed to be vascular pathogens, that they're predisposed to be Xanthomonas oryzae type pathogens, and that the TAL effectors that we've tested so far do not explain tissue specificity of the pathogens. So we now know a little bit more about the biology of this organism. But I'd like to conclude by talking about, can we use this U.S. Xanthomonas oryzae strain as a tool to assess TAL effector function in plants? So again, I'll remind you that they both have large numbers of TAL effectors. In fact, oryzicola has over 26, and we know nothing about the function. And in fact, it's been shown for many years that there's no one has identified single gene resistance to Xanthomonas oryzicola. So we need sources of resistance. So why aren't we seeing resistance with these TAL effectors? Well, it could be that the expression of multiple TAL effectors is suppressing the activity of an individual TAL, and you won't see it. So we wanted to ask if we could use the US strain as a platform to assess the roles of virulence of these TAL effectors in diverse rice hosts. So we started out with the oryza SNP set because it's very well characterized, and this is a set that Ken McNally led a project on a number of years ago to obtain resequencing for the genomes of these various organisms, and you could of these various rice plants, and you can see that they have a huge diversity of phenotypes. And we know them to have a huge diversity of responses to the bacterial pathogens. So Renee and Valerie and Lindsay inoculated these plants in multiple experiments over multiple times and asked the question, what happens if we inoculate these 20 varieties with the US strain X115A harboring different effectors? So AVRXA7, PTHX01, and TAL-C in this case. These are different TAL effectors. And so you can see here on this very difficult to interpret graph that some of these TAL effectors indeed did, as we expected, enhanced the virulence of the US strain. So the clear bars are the US strain on its own, and you can see hugely enhanced virulence by the addition of these TALs. And we actually looked at the relationships of the enhanced virulence in these this genetic population. And so, for example, most of the japonicas enhanced we saw enhanced susceptibility with any of the 3 tals that we tested. Some of this makes sense because in some of these indica lines where we saw maybe 1 or 2 tals inducing resistance, these populations have been bred to contain resistance in some cases. But in some cases we actually saw decreases in the lesion lengths, and in fact, before I come to that, I want to tell you that we also monitored the expression of the target genes and asked the question, so these are the OS-suite genes, and we asked if we inoculate with the US strain containing the different tal onto these particular different varieties of rice, and there's a subset of the varieties, what do we see? Do we see the activation of the susceptibility gene? And indeed we do. But what's interesting is we do not see a correlation with the amount of activation of the gene and the amount of virulence. So there could be a number of reasons for this. So while the target activation was consistent with the activity, and the promoter sequences didn't vary, Lindsay went in and sequenced the promoters from the OS-suite genes from these particular varieties, and they didn't vary, the activation didn't correlate. So our hypothesis is that there could be other targets for individual tal in the genome. So what that means is that they're not just activating one gene, they're possibly activating a number of genes. So if you're thinking of engineering rice to respond to a particular tal, you need to think about what other things it's activating. So that's number one. There are also there could be differences in the OS-suite coding regions, which we didn't check. And there also could be differences in the genes that modulate the suite activity downstream of the tal effector activation. So we've opened up a new set of biology to study in terms of the tal effectors and their interactions. But I want to come back now to, can we use these tal effectors to screen for virulence? So in our 20 varieties, we had inoculated with X11-5-beta-A containing the AVRXA7 gene, and we asked, do we see induction of a dark browning response, which is consistent with a hypersensitive response? And we know AVRXA7, when in combination with the host resistance gene XA7, does give a dark brown or a hypersensitive type response. And sure enough, and I'm going to blow that section up, in 2 varieties, if you look at this plus, it indicates that the AVRXA7 gene, we're able to identify single resistance genes. Now, of course, this gene is known, but the question now will be, can we identify novel sources of resistance? And in fact, we have seen hypersensitive type responses in cases where we really don't know what the interaction is. And so that's the next steps for the study. So today I've told you a little bit about the biology of tal effectors and how they interact with plants to cause disease. Some of the tal effectors increase virulence of the US strains, but they don't convert XOC to XOO, and the XOC tal effectors don't convert the US strain to Xanthomonas oryzae or to a vascular pathogen. The tal effectors also increase the susceptibility to numerous varieties, as we expected. They're virulence factors, and that's why the host wants to recognize them and block the ability, which confirms that the suite targeting tal effectors are major contributors to virulence. And finally, the individual tal effector activation of resistance can be studied using these individually cloned tal on a wide variety of rice cultivars. And in fact, Valerie and the student Renee have already started those screens on very diverse populations. It's a very simple screen. So I'd like to thank those who did the work, including Valerie, who is here at the meeting, and you can chat with her more if you like. Renee Carell is a student, was an undergraduate at the time and is now a graduate student in the lab. Lindsay Triplett, who did a lot of the molecular assays and the sequencing of the strains, and Paul Langley, who helped a lot with the experiments in undergraduate. And also our very wonderful collaborators in Adam Bogdanov's lab, and Adam was at Iowa State when these experiments were done, and he just recently moved to Cornell. And finally, our funding agencies below. So with that, I'd be happy to take any questions. If there's time.
Thank you very much, John, for the excellent presentation. So, not so much times we have, but only one or two questions.